Hello, this is Ms. Hunt again, and I'm finishing up my Chapter 11 lecture on cell communication. So let me put this in presentation. All right, and let's get started. This is where we left off, and we were talking about signal reception, transduction, and responses. So we're going to continue on here. In reception, the target cell directs the signaling molecule that binds to the receptor protein on the cell surface. So I drew you up a nice picture, and I said in the reception right here, that target cell is going to be binding to the receptor protein on that cell surface. In transduction, the binding of the signal molecule alters the receptor and initiates the signal transduction pathway. So that's when we hit and then we have it passed down going through the transduction pathway. In response, the transduced signal triggers a specific response in this target cell. So here we go. Here is our phospholipid bilayer. Here is our receptor protein. Our signaling molecules coming in. So this is reception. This is the transduction, that relay of passing it down through the cytoplasm and then receiving it, an activation of a cellular response. All right, here it is again. This is a great little clip just to show that animation, okay? The receptor's taking it in, we're transducing it, and then it's coming into being received. All right, 11.2. Reception, a signaling molecule binds to a receptor protein, causing it to change its shape. So this is very similar to like an enzyme where it binds and then changes shape. The binding between a signal molecule, ligand, and a receptor is highly specific. So they're going to call the signal molecule right here a ligand, and they're always going to call it that ligand. So here's your receptor. I said it kind of looks like a letter Y. And the ligand is what is binding to that receptor. A shape change in the receptor is often the initial transduction. So the first thing that happens, the receptor changes shape. So when the ligand hits, and then this receptor changes its shape, or it allows it to come in, but the shape changes in the receptor. Most signal receptors are plasma membrane proteins. The G protein coupled receptors, they call them GPCRs, and you're going to see this several times, are the largest family of cell surface receptors. So ligand is what binds, okay, a signaling molecule. And one of the types of receptors is the GPCR, the G protein. Most water-soluble signal molecules bind to specific sites on the receptor protein that span the plasma membrane. So some people are born with disabilities where they are missing certain protein receptors that are throughout that cell membrane, and that causes different problems throughout the body. You probably have all your receptor proteins, and that's why you're functioning as well as you are. There's that ligand, that signaling molecule that comes in to our GPCR, and it's taking it in. And then there's lots of different pathways that it can come through to do something with. Here's another 3D model of it, okay? There are three main types of membrane receptors, and yes, you do have to memorize this. There are G proteins, there are receptor tyrosine kinase, and there are ion channel receptors. We've talked about ion channels before, that they have a charge that goes through them, okay? So here's three different types of receptors that are coming through here. Receptor protein, so that's what we're talking about in the cell membrane, a receptor protein. The G protein coupled receptors, the GPCR, are cell surface transmembrane receptors that work and help of a G protein. Okay. The receptor tyrosine kinase are membrane receptors that transfer phosphate groups from ATP to another protein. So that's pretty important. Every time the ATP loses that P, the phosphate, to be able to become ADP when it's popping off to give energy, that phosphate still needs to be taken somewhere and done something with. So that the receptor here is part of that process. The receptor tyrosine kinase can trigger multiple signal transduction pathways at once. Abnormal functions of the RTK is associated with many types of cancers. So when this doesn't work, it leads to different kinds of cancers. Okay, there's the G protein we've talked about. Okay. All right, I was looking for the tyrosine, but it didn't show it, the RTK on there. Okay, but it is doing the G protein being activated. Okay. All right. The last one was that ion channel, and they call it a ligand-gated ion channel. Is a receptor that acts as a gate that opens and closes when the receptor changes shape. When a signal molecule binds as a ligand to the receptor, the gate allows specific ions such as sodium and calcium through the ion or through a channel in the receptor. Remember, ions usually have charges, or ions always have charges. And these are going to, so that's the top view, the open and close, so it's changing shape. So this is that ion channel when the ligand binds and it's coming through. Okay, so here is the signaling molecule, the ligand. The ligand is binding. This is that tyrosine that's on here. And you can see that the tyrosine is activated and ATP pops off. A fully activated receptor, tyrosine kinase. That's what was the previous one, where the phosphate is going to be popped off on here. Okay, there's our tyrosine kinase. The ATP and the phosphates are popped off. All right. And it looks like we don't have a picture for the ion, but it's very similar that you know the sodium and the chlorine or calcium are going to be popping through. They have a charge. It's going to bind to the receptor molecule and move through. Okay. Intracellular receptors, so inside the cell, intracellular receptor proteins are found in the cytoplasm or nucleus of target cells. They're small or hydrophobic chemical messengers can readily cross the membrane and activate receptors. Examples are hydrophobic messengers are steroids and thyroid hormones of animals. Remember we said the thyroid was right here? An activated hormone receptor complex can act as a transcription factor, turning on and turning off specific genes. So remember, this is all about cell communication. How that hormone is sent from that thyroid gland to all throughout the bloodstream and the endocrine system to everywhere. This is saying that that hormone is going to, remember hormones and steroids are fats, so they're going to pass through the cell membrane. They would be hydrophobic because they're a lipid. They'll pass through the cell membrane and then they're going to bind to something in the cell, in that intracellular. In honors biology, we talked about transcription and translation of building that protein. We're actually talking about it right now in honors biology. If you guys remember, the DNA in the nucleus is opened. The mRNA with the RNA primase gets coded. The mRNA comes out. The mRNA goes down to the ribosome, and it's read with the codons matching the anticodons that drop off specific amino acids. And this makes the protein. So in this type of intracellular signaling, something is traveling in. This is actually really awesome. Right now, Pfizer has made a vaccine for COVID, and they're making an mRNA. And you're thinking, well, how is an mRNA going to work? Well, exactly right here. The mRNA from our vaccine won't go to the nucleus, but it is going to go to a ribosome. The ribosome is going to build a protein that's going to start the immune system to fight off a COVID virus if it comes into our body. And it's going to stop it from allowing it to come into our body. So Pfizer has actually built a sequence of mRNA, which is pretty awesome. And it's going to, code for a specific protein inside. So that's pretty cool. But this is transcription and translation. So whatever is being built for us who are talking about for our signaling, the signal comes into the cell, whether it comes in the phospholipid bilayer through a protein or not. If it's coming in, and this part is the intracellular part, it's coming straight to the nucleus. All right, a code is being asked to be created. So that's going to trigger transcription. Transcription will build the mRNA, come out for translation, go to the ribosome, and build the protein that's needed to do whatever your body is coding for it to do, signaling for it to do. Okay, here is a hormone. I'm trying to remember how to say it. Aldosterone. What it does, I have a link right here. It keeps the salt in your water at a constant amount. So here's the little link for it. I have it right up here too. Here we go. It's a steroid hormone, so steroids are fats. That is main role is to regulate salts and water in the body, thus having an effect on blood pressure. So that's pretty cool. So here is the hormone who's coming in. It comes right in through the phospholipid bilayer. It doesn't need a transport protein. The hormone is going to bind to a receptor protein inside the cell, which triggers it to come to the nucleus. You see them both coming into the nucleus. Now we have transcription happening. It's finding the piece of DNA that it needs to build the code. It makes the mRNA, mRNA leaves the nucleus and travels to the endoplasmic reticulum, where the ribosomes build the new protein that is needed. And that protein will help keep your blood pressure level and keep the right amount of salt in your bloodstream. Here it is again. Transduction, that's passing it down. Cascades of molecular interactions relay signals from receptors to target molecules in the cell. Cell signaling is usually a multi-step. Multi-step pathways can greatly amplify a signal. Multi-step pathways provide more opportunities for coordination and regulation of cell response. The binding of a single signaling molecule to a receptor triggers the first step in the chain of reactions. The receptor activates another protein, which activates another, and so on, until the protein produced, the response is activated. Excuse me. At each step, the signal is transduced into a different form, usually a shape change in a protein. Protein phosphorylation and dephosphorylation, remember that's the phosphate popping on and off, of proteins is widely cellular mechanism for regulating protein activity. Protein kinase transfers phosphates from ATP protein in a process called phosphorylation. We've heard this word many times already. Okay. And may relay molecules of a signal transduction pathway, creating a phosphorylation cascade. So here's the receptor protein, the signal coming in, and this is what they call the cascade. So you can see it coming down, coming to the next, and we get a cellular response. This is the phosphorylation cascade coming down. And dephosphorylation removes the phosphate from the protein. Okay, seeing how much further I have. Okay, I'm going to stop here because I have to go cover for another class. So we're going to stop here at dephosphorylation, and I'm going to make my third video probably right after school today.